for our discussion of vertebrate diversity, we're going to look at a couple of major trends across all vertebrates, and then we're going to focus in on just one major group, the snakes, as an example of some of the things that can happen to create new body forms. Now remember, review the chordates. So chordate is a large group, and vertebrates are by far the most important diverse group within the chordates. But the chordates also include the cephalochordates, which are really just a remnant group, once much diverse in the early history of the Paleozoic era, but now just kind of a remnant. The Amphioxus or lancet are the remaining species. And then also we have the urochordates. The urochordates are sometimes called tunicates or sea squirts, and although they don't look much like chordates when they are adults, as juveniles they have all the typical chordate features. So just take a look at those and review those, but we are going to focus on the vertebrates. So the vertebrates include some really diverse and important groups, including mammals and reptiles. Remember, birds are part of the reptiles. Of course, the fish, which are a very, very diverse group. Okay, and one of the things that sets them apart is this diversification, differentiation on the anterior to posterior axis. So we're going to use the higher vertebrates, which includes, we'll call them the tetrapod vertebrates, right? That's the group we're going to focus in on. And tetrapod vertebrates include amphibians, reptiles, remember throw birds in there, and the mammals. And they have this unique diversification of different regions of their vertebral column. So if we look at cervical vertebrae, I've flipped this around so the writing's backwards, but the cervical vertebrae of a human are very different in form than the thoracic vertebra. And we've got this attachment for the ribs, and of course ribs are now articulated with the thoracic vertebra. And then we can look at the lumbar vertebra, the sacral vertebra. So there are differences in all of these different body regions. Okay, and so as we're looking at this, we need to come up with general rules. And one of the things that we see in vertebrates, as mentioned in a previous class, is that in mammals, there's a very, very conserved, there are only a couple exceptions to this rule, but there's a very conserved rule as to where the cervical vertebra end, or in other words, where the neck ends, and where the thorax or the thoracic vertebra begin. So whether you're a giraffe with an incredibly long neck, a whale with almost no neck, right? It's hard to see from the outside, but whales do have a neck. Or a human or any other mammal, you have 7 cervical vertebrae. So that's a very, very conserved pattern. I think there's a single species or maybe 2 species of sloth that break that mammal rule, but it does not seem to be very flexible. Whereas in other groups, there's tons of flexibility. So in the reptiles, again, including the birds, you can have a couple dozen, I think 30. I want to make, actually I don't know. Let's look it up. That's why we have the internet, right? So let's look it up. A reptile with the most vertebrae. Let's see if we can find an answer here. I don't know, maybe it's in this osteology of vertebrates. Kind of a weird question, but anyway, I was curious, but it looks like it may not be all that easy to answer. So I think that some of the, oh, maybe because I need to put reptile with the most cervical vertebra. One last shot. No, internet, you can tell us anything, right? Yeah, lizards, white-owl mammals. This is an interesting one, but all right. Anyway, some of the ducks and geese can have, 10, 15, 20, sometimes even more vertebrae. So the point is that amongst the reptiles, the vertebra number is incredibly variable. They can have a few vertebrae or they can have many vertebrae. So that seems to be a much more flexible and dynamic thing across the history of the vertebrates. And then of course some just have like basically a single cervical vertebra, like frog. This is not a reptile, but the early vertebrates, the amphibians, can have a single vertebrate. So there's lots and lots of diversity, except for mammals, which are locked into this kind of 7 vertebra ground plan. Now, when we look at mammals and reptiles, we see an incredibly similar pattern in the genetics of how the barrier between the neck and the thorax is defined. So this is basically the end of the cervical vertebra, and it has to do with a Hox gene. So Hox C6. Don't worry about the other Hox genes. They play roles also in patterning different regions, but we're focusing just on Hox C6. So where that boundary is, that defines the transition from the cervical vertebra in green to the thoracic vertebra in pink. And even if we have, what is this, like 14 vertebra in the chicken, cervical vertebra in the chicken, and we've got something like 20, I guess, 18 in the goose, that it's still defined by that same transition point of the Hox C6 gene. So just memorize that. That's an important concept. And again, mammals, it's always at the same part. Now notice it's somite number 12. Remember we talked about somite formation and how we go from a wave of expression of notch at the anterior end, and then it moves all the way to the end and forms these. And so we number them from 1 to however many at the end, and there's variations in the total number also. Okay, but Hox C6 defines the boundary between the neck and the thorax. Okay, other Hox genes do other more complex things. We're only going to focus on this one as an example. Okay, and so when we look at snakes, at a python, they can have hundreds of basically thoracic vertebrae. So if you look at a snake, the kind of, I guess, initial response for most people is they're all neck, right? You've got a head and then a neck going all the way back. But if you look at it, that actually doesn't make sense, because the bones of snakes, individual bones here, as we show here, look way more like thoracic vertebrae, right? So they've got these attachments and ribs. Even the ones at the front have these little projections coming off that look just like ribs. So rather than being all neck, snakes really are all thorax. This transition, this anterior boundary of the Hox C6 expression, has shifted all the way up basically to the base of the skull. So in essence, we have lost the neck in snakes, and the thorax has shifted all the way up. Now, in addition to that, there are other interesting stories too. So in addition to just being all thoracic vertebrae, then they also later lose their legs. So that's another transition and a different story with different genes. Okay, but we can look at this Hox C6 expression and see it a very posterior. Here's the head, here's the neck, and there's the beginning of the Hox C6 in the chickens. We have this long area that developmentally is going to be the neck, and then in the snake, this is shifted all the way to the anterior region. Okay, so no Hox C6. Know that snakes are just basically all thorax. We've completely lost the neck in snakes. And so this brings up a very interesting idea of how we change the expression domain, the region where genes are expressed. And it turns out that there are basically two ways we can do this. So let's define the two ways we could change the domain. Okay, and it's got to come back to a change in expression. So we're going to look at transcription factors. Now remember, the early stages of development, we're talking transcription factor 1, which then has an impact and turns on transcription factor 2, and then so forth. So these are all transcription factors early on. And if we wanted to change where a gene was expressed, we could either change the transcription factor that is targeting that gene, or we could change this regulatory element so it's no longer able to bind the things that were turning it on before. And both of those, a change in the upstream transcription factor or a change in the regulatory element that binds that transcription factor. Those are the two things. So you got this list here. I guess it's in the outline. The two things. Number 1, change in transcription factor. Number 2, change in the regulatory element that binds that transcription factor. And so when this happens, we could, in essence, by changing regulatory elements on Hox complex A, we could change where 9 was expressed. And so even though they might have conserved ancestral feature, they can evolve new features by this change in domain. And they may even maintain the exact same function. And so most of the time, it's those regulatory regions that change. But you can have an upstream regulatory region that changes and then has a downstream effect on the next gene in line, or 2 or 3 genes later. And so that regulatory evolution really is key for evolution of organisms. And now we can actually study those. And so we can look at the upstream areas of a mouse, a whale, and a chicken, right? We'd expect the mouse and the whale to be fairly conserved. So on this diagram, everywhere you have a dot, there's a difference. And everywhere you have a dash, everywhere there's a dot, it's the same. If we have a dash, that means we've eliminated some DNA sequences, and the letters represent differences. So notice that we can define a reptile-specific motif here in this regulatory area. And then there is a whale-specific one, and so then we would look at these genes. This is one that controls HOXC8, and we look at the difference between where these things are expressed. And so we actually see this transfer, this posterior expression in the whale that is defined by this one regulatory element. The rest, if you look at the whale and the mouse, everything else is identical in the regulatory region of this gene. And so now we can very, very specifically point to single changes. And I don't care that you memorize this one single change, but just know that by sequencing these DNAs and then putting them in a comparative context phylogenetically, we can look at where they're expressed.